Good afternoon, everyone. We are at week 11 this week, so just 4 more weeks to go, 3 more weeks of lecture, and then the final week is going to be test number two. So as we wrapped up with the semester, we have 3 more lessons to go, and today we will be doing lecture number 8. So let's get started. Number 8 is about heredity, specifically how genetic information are passed on to the next generations. The first half of the lecture will be theory-based, and then the second half of it will be application problem-solving based. We will begin our discussion with the concept of meiosis. So you did the quiz last week, which means you should be pretty familiar with the process of mitosis. And so now we will learn about meiosis, which is fundamentally different from mitosis. But I will highlight a few key differences for you as we go through the slides. Okay. First of all, the purpose of meiosis is to enable sexual reproduction. So sexual reproduction involves two individuals. There are reproductive cells from each parent, and when they join, they are going to create a baby. And the basis of creating these reproductive cells involves the process of meiosis. Critically, the process of meiosis actually causes the number of chromosomes to be reduced by half when it is producing these reproductive cells. So there are some key and new terms that we should learn about. Haploid and diploid, for instance. Humans are diploid organisms. Diploid means each cell has two sets of chromosomes. So we know we have 23 chromosomes from each parent, and that gives us two full sets. We say 2n. Okay? 2n equals to 64. Right? So this number at the front tells you the number of set of chromosome you have, and then this n number is number of chromosomes per set. So in our case, each set is 23 chromosomes, and we have two full sets of it. That makes us diploid. Right? So our somatic cells, which are bodily cells, right? So that's everything except for the sperm and egg. They are going to have two sets of chromosomes. They are all diploid. Human gametes, which are sperm and egg, they are haploid. They only have one set of chromosome, so we call them N instead of 2N. And so when a sperm fertilizes an egg, then it's N plus N, which gives you back the 2N, right? Each parent contribute one full set of chromosomes, and it's passed on to the next generation through these gametes. Okay. So to illustrate this further, we have haploid cells. In this case, n equals to 3. There are only 3 chromosomes in the set. A diploid cell would be 2n equals to 6, total of 6 chromosomes with 2 full sets. Each set consists of 3 individual chromosomes. If it has 3 full sets, then we say triploid, okay, or 3n. In this case, 3n equals to 18. So in all these examples, the n number does not change. The number of chromosomes in one set does not change. What's changing is how many sets you have in the cell. It's possible for cells to be tetraploid as well. So 4n equals to Sorry, I made a mistake. This is 3 times 3 should be 9 here. And here, 4 times 3 is going to be 12, right? So again, the n number does not change, but the number of sets and the total number of chromosomes are going to be different. So for strawberries, they are actually octuploid, 8 sets. And then for this is sweet potato, or some people call it yam, it is a hexaploid, right? So whenever you have more than 2 sets, we call them polyploidy, polyploidy, which is more than 2 sets of chromosomes. And you can specifically call them triploid, tetraploid, so on and so forth, but generally, polyploidy, it's more than 2 sets. Okay. Now, one of the questions I had, growing up was how do you get seedless watermelon? To grow a plant, you need a seed, but how do you grow a plant that has no seed? Well, it turns out the answer is quite interesting. Regular watermelons are diploid. That's the kind of watermelon that has, the black seeds in them, right? Those are diploid, two full sets of chromosomes. And then they use a special breeding technique, which we're not going to go into, but that creates tetraploid watermelon. So these are watermelons that have 4 sets of chromosomes. Okay. So I want to ask you, based on everything we talked about so far, for the regular watermelon, their gametes, plants also have gametes. We just don't call them sperms. Okay. We call them like pollens, right? Pollens and eggs. Right. So for the gametes of a diploid watermelon, it is going to be N, right? It's haploid. But what about for the tetraploid watermelon? What would be the number of N's for something that is tetraploid? Anybody? That's correct. It is 2N, right? We basically divide this number by 2, right? And so if these things come together and fertilize the egg, N plus 2N gives us 3N. So this 3N here is the seedless watermelon. It turns out if something has an odd number of sets of chromosomes, then it is incapable of reproducing sexually. They are sterile, they cannot produce seed. And the reason is quite simple. If you take 3n and you try to divide by 2, it's not a whole number, it's 1.5. You can't have gametes that are carrying only 1.5 sets of chromosome, right? You need the full set or multiples of a full set, right? So the key thing to note from this example is well, not only do we now know where seedless watermelon comes from, but we also know cells with odd numbers, odd number set of chromosomes cannot produce gametes. Okay, so no gametes means no sexual reproduction, no seeds. Are we together? All right. So for sexual reproduction, begins with meiosis in germ cells. Okay, so what are germ cells? I sort of told you this before, but germ cells, basically, it says right here, germ cells are diploid reproductive cells, 2Ns, that give rise to haploid gametes by meiosis. So in other words, you start with a germ cell. They have different names for male and female. We'll get to that later. And it's going to go through 2 rounds of divisions. Okay? And in the end, you get cells that are only going to have 1 set of chromosome. Okay, so these could be like sperm or something like that. Okay, so where does it happen? Well, in male, it happens in the testis, and then for Females, it happens in the ovaries. So these reproductive organs, they are called gonads. Gonads are reproductive organs. Okay. So to put it in a full sentence, I guess, the germ cells are found in the gonads and they undergo meiosis, which give rise to gametes. Okay. We'll learn about the reproductive system next semester. So the egg gets released from the ovaries, and it's going to enter the fallopian tube. Um, Fallopian tube, or also called the oviduct, and that is the location where fertilization could take place. So if sperm is available, it will meet the egg inside the fallopian tube, and once the egg gets fertilized, then we have a zygote. A zygote is the first cell that you get after the union of the sperm and the egg, and this is diploid, 2N. And then the zygote will rapidly divide in the coming days following fertilization, and eventually it will become the embryo and in turns develop into the fetus or the baby. So here is an overview of meiosis rather, sorry, meiosis. We have 2N, and after DNA replication, it is still 2N, and then the cell would divide. And in this case, because these are still considered as like the same chromosome stuck together, so even after the first division, it's already considered as haploid. And then when you divide again, it is still going to be haploid, okay? Because we just count, it's the way we count the chromosomes, right? Basically, this is considered as one chromosome, and so is this, right? So that can be a little bit confusing, but the key thing to remember is you start with diploid, and then after one division, it's haploid, and then if you divide again, it is still haploid. So the meiosis process, it's all written out here. So I'm not going to read through the slides with you, but I will I will go take a note with you that compare mitosis and meiosis to help you highlight some of the key differences. This table that I'm about to fill out is in your study guide. So please take that out if you have it. I will just copy it and paste it over if it lets me. Nope, looks really bad. Just give me a second. You know what? I'm just gonna copy half of the chart first, and then I'll bring the other half over in a bit. All right, there we go. So let's start with, by the way, we'll use human as a example, okay? Humans. All right, let's start with the mitosis. Number of cell division. How many times does the cell divide in mitosis? So let's say you start with one cell. How many times does this cell divide in mitosis? That's right, it only divides once. So you're gonna basically end up having two daughter cells. Two daughter cells. Okay? So one division. Once. One division, we can say. Now for meiosis, as you just seen in the um, slides, you actually will get two divisions. So you end up having four cells. There we go. Two divisions. Number of daughter cells produced. Well, you can tell from the picture, you'll get two daughter cells. Two daughter cells. For meiosis, you can tell from the picture you get four cells, right? But actually, it's a little bit different for male and female, and we're going to talk about that difference in a bit. For female, they will get one egg plus three polar bodies. I will explain what that means later. Okay, just copy it down first. And then for male, each meiosis will produce four sperm. Okay, the daughter cells that we get for mitosis, are they diploid or are they haploid? Do they have two sets of chromosomes or just one set? That's right. If you have been following along, you start with 2N and afterwards it is still 2N, two sets, diploid. 2N equals to 46. And for meiosis, you start with 2N. After one division, it's N. And then you divide again, it is still N. And again, I know it's confusing, right? Like don't use math to think about this. Like you think 2N divided by 2 is N, and then N divided by 2 should be like N over 2 or something, but no, it's not quite like that, okay? It has to do with the way we count the chromosome, right? The key thing is for meiosis, what you're going to get in the end, it's haploid. N equals 23. Okay. Number of chromosomes. Oh, I answered my own question. I'm sorry. Sometimes I get ahead of myself. Here, I'm just gonna move these over. There we go. All right, so that's like rudimentary information. Right, nothing too complicated. Now we're gonna compare prophase and prophase 1. And again, you did the mitosis test. Actually, the class average was like very, very high. So I guess you guys all had a good understanding of mitosis. So this should not be too hard. Okay, we're gonna have a cell here, and I'm gonna use red to represent the maternal chromosome. Okay, remember the chromosomes are a number based on size. So these really big ones are chromosome 1, and then these really these smaller ones, they are chromosome 2, and of course it goes to 3, 4, 5, 6, all the way to 23, right? So in prophase of mitosis, by the way, like what do we call these chromosomes? I just want to make sure you guys are with me. Like this big one here, 1, 1, 1, 1. What's the name we give for the ones here and the ones here, the blue and the red? Does anybody remember? Oh yeah, maybe my question is not specific. So like this, the red and the red, that's sister chromatid. Yeah. But the red and the blue, it's homologous, right? So homologous chromosomes are randomly scattered, I guess, or like a better word, throughout the cell. Okay. They're just here and there, all over the place, not really, you know, have any patterns or organization to it. And then in prophase one, I probably should clarify this in case, because sometimes, like, I think it's obvious, but then actually it's not. Meiosis has two divisions, right? So we have meiosis 1 and meiosis 2. And under meiosis 1, we have prophase, metaphase, anaphase, telophase 1. Okay, and then here we have prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, so the names are same as the mitosis. We just separated it into 1 and 2. So over here, we are specifically looking at prophase number 1, the first round of division and the beginning of it, basically. Okay, I actually, you know what? I think I might be going all over the place here. Let me just pause for a second, let you copy whatever you need. Okay. So in prophase number 1, this is what happens. Chromosome 1 from mom is going to find chromosome 1 from dad, and they're going to pair up. Similarly, chromosome 2 from mom will find chromosome 2 from dad and pair up. Okay? So homologous chromosomes pair up to form tetrads. Okay? To form a tetrad, I guess we can say. To form a tetrad. Tetrad means 4, right? So one, two, three, four sister chromatids stuck together. That's a tetrad. Okay, and then here we have another tetrad. So you're gonna have a total of 23 tetrads, right? Unless it's male, then the X and the Y's don't really come together. But that's okay. Like generally, the homologous chromosomes will form a tetrad. And the point of doing that is allow something called crossing over occurs between homologous chromosomes. This is too thick. To create genetic variation. I'll explain. Just copy it down first. Okay, so here is the mother chromosome 1, And this is the father chromosome 1. And remember, on these chromosomes, they actually consist of the same kind of genetic information, just not necessarily exactly the same. I told you this before, right? Over here, it might be like eye color, and it's going to be eye color here as well. But one might be blue eyes, one might be black, brown eyes, right? Depending on who your parents are. On a different location, like here, it might be you know, type of hairline, right? Is it straight hairline or is it a widow's peak, right? Again, it depends on who your parents are. So what happens is the homologous chromosomes can come together and then they would literally intersect with each other. That's what we call a crossing over. Crossing over. Now, I am obviously oversimplifying the situation here. You're not just going to have like two crossing over. You're going to have multiple crossing over along the entire chromosome. And afterwards, what happens is basically the red and the blue kind of switch places. And in the end, you're going to have, um, the red chromosome that has swapped a little bit with the blue. And then similarly, this is really hard to draw, sorry. And similarly, the blue is going to have a little bit of red, right? So this here is called genetic recombination. Okay? Essentially, you are swapping the genes on the mother chromosome and the father chromosome. Now, I know that's a little bit confusing. When I say mother chromosome and father chromosome, I'm talking about the chromosomes you inherited from your mother and your father, which is inside your cells now, right? So then the chromosomes get swapped, shuffled, and then it gets passed on to the next generation. Okay? Now, again, this is oversimplification. I'm only showing one part of the red and the blue get exchanged, but in reality, it's happening like all over the chromosome, right? So it's more like, don't draw this, but it's more like, that, and then you're gonna fill in the gaps with the blue, right? And the same thing for the other one, right? So essentially, you reshuffle the deck before you pass it on to the next generation. Does anybody have any question about what the purpose of this is and how it's happening? If we don't do this, then we end up passing on uh, the exact chromosome onto the next generation, and there will be very limited variation in the population, and that's not a good thing. Okay, so this situation, this crossing over, happens in prophase one, but it does not happen in prophase. Okay, you don't want it to happen in prophase of mitosis. In mitosis, you want a faithful copy of the original cell. Okay, moving on. If nobody has anything they want to ask. Next is going to be metaphase of mitosis, which again, you already know. Okay, we're going to have some centrioles here. Okay, and then the chromosome is going to line up in the middle. And you can draw the spindle fibers if you like. Okay, so chromosomes lined up at the center of the cell. Okay. Again, you should know this already. Nothing's new. Pretty standard. And then for metaphase 1, which is the first half of the division in meiosis. You will also have the centrioles. And this time, we're going to have the chromosome, sorry, the tetrad lining up in the center instead of individual chromosomes. Now, this part is a little bit hard to draw, but remember the crossing over has already taken place, and so there's a bit of blue on the red, and then there's a bit of the red on the blue. It's going to be all over the place actually, but it's easier to just show a little bit of it, right? So instead of chromosomes lining up at the center of the cell, we will have tetrad. Tetrad. Tetrads line up. Any questions? Are we together? Okay, thank you. All right, moving on. Next, we have anaphase of mitosis versus anaphase 1. Let's take a look at anaphase. Which I'm sure you still remember quite well as you just did your quiz. Can you tell me what these little red lines are? What do we call them? I mean, I know they're chromosomes, but like specifically in this context, what should we call them? Something are separated. Yes, that's the word. That's the term I'm looking for. Okay, sister chromatids. Sister chromatids are separated and are pulled to the opposite ends of the cell. Okay. And over here, oh, I just remember I forgot something. Um, Give me 1 second. Let's finish this first, and I'm gonna add to it. Okay. So I'm gonna use the same description. Oops. Over here. Oh, sorry. This ends up like even more work than just writing it, I suppose. Okay, so here's something I separated and pulled to the opposite ends of the cell. What's the word here? What should I write in the blank? In mitosis, the sister chromatids, and over here. Okay, guess Elvis. Okay, Elvis got all the answers, which is good. These are homologous chromosomes. Okay, the rest of you okay? Do you understand the terms? All right. Now, I missed something here in metaphase. So just gotta give you a moment to catch up, and then I'll add that extra bit. I'll do it in green, nice catchy color. Okay, so the orientation. Orientation is like, the positioning, okay? The orientation of each tetrad is random and independent of each other. This further increases genetic variation. Okay, so what does that mean, right? Over here in the first tetrad, you see the blue one is on top and the red one is at the bottom. And then the next pair, red one is on top and blue is at the bottom. And remember, we have like 23 of these tetrads, right? So, and then the next one, it could be red on top or blue on top. And then the next one, again, like you have two choices, right? So how you position these tetrads, whether it's the blue copy on top or the red copy on top, this is completely random. And how one pair position itself has no effect on how the next pair position itself. So it's like flipping a coin 23 times. The result of each coin, right, heads or tails, or like blue on top or red on top, has nothing to do with the next pair. So this will give us 2 to the exponent of 23 additional combinations. Okay, because it's two choices for the first one, blue on top or red on top, and then two choices for the next one, all the way up to the XX or XY at the end. And so that further increases the genetic variations. Right? You never know which one's going to get passed on to the next generation. Okay. Now telophase is just going to be the same as regular telophase, nothing to write there. And the genetic relationship of the daughter cells to the parent cells, well, for mitosis, it is identical. And for meiosis, it's not identical. Right? After all that gene shuffling, they are going to be different. Not to mention they're haploid. You only get half the genetic information. Type of cells involved, somatic cells. These are bodily cells. And over here, these are germ cells. Okay. The gametes, the sperm and the eggs, they are the end product. Sperms don't produce more sperms. Eggs don't produce more eggs. The germ cells will create more sperms or more eggs by undergoing meiosis. Why do we do mitosis? Growth and development. We have many more cells now than we once did. We all began with a single cell called a zygote. Now we have trillions of cells. How do we get so many cells? Most of it is from mitosis. Replace lost or damaged cells. We do not use mitosis for asexual reproduction, but some organisms do, right? Like amoeba. For sexual reproduction, sorry, for meiosis, I gave the answer away. For meiosis, the function is to produce gametes, okay, which is sperm and eggs in humans for sexual reproduction. All right, that's it. That's the comparison between mitosis and meiosis. Anybody want to ask me anything before we move on? Thank you. All right, that's that. Let's go back to the PowerPoint slides. You can read through the description yourself, but just look at the picture here. Right, we have the tetrad forming, and you can see crossing over happening between the homologous chromosome. Okay, there is no crossing over between the sister chromatids. It's like there's no point. They are the same, right? So like the blue one's not gonna cross over with the blue one. You're just gonna end up swapping the same genes. It's like no difference. But you will do it with the homologous chromosome. I know sometimes they say centriosome. Don't worry about it. Just change it back to centrioles, okay? For us, it means the same thing. Okay. Then in the metaphase, we have the tetrads lining up in the middle. Again, the orientation is random. It could be blue on top or red at the bottom, or red on top and blue at the bottom. It's totally random. And then in anaphase, you separate the homologous chromosomes. And then the next step, you're going to basically split the cell in half, and that's going to be one cell. That's going to be another cell. So like this. All right. Now let's go back here for a second. Uh, so over here, how many chromosomes does this thing have? Well, if you count it, that's like one, two, and then three, four, right? So that's four chromosomes in total, and that is two sets. So 2n equals to 4, two sets. Okay. Uh, blue, blue, that's one set, and then purple, purple, that's another set. Now fast forward to over here in telophase. Okay. Now how many chromosomes do we have? One chromosome, two chromosomes, two chromosomes. And that's just one set. Right? One big size, one small size, one set. That's why after the first division, it's already haploid, just N. Okay? Same thing here, N. And
I
then,
have a question.
yes, go ahead.
Can we still say that we have two sister chromatids?
Can you still call these sister chromatids? Is that your question?
Yes.
Yeah, that's a tricky one, okay? Technically, no, because after genetic recombination, they are no longer identical, right? Okay, but they're still stuck together, so it's still okay to call them sister chromatids, okay? But just like in a different context, right? If you would. Does that answer your question?
Thank you.
All right, and then we have the second round division, prophase, which is now all same as mitosis, okay? They're gonna randomly align, and then they're gonna go to the middle. You separate the sister chromatids, even though they are not exactly identical, and then you split the cells. So you end up having four different cells. And again, each one has a big one and a small one. That's N, 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 and then N. Okay. So I'm not going to ask you to describe the process, but it's it's good to, watch the animation I post on the video resources. But for the purpose of the final test or the quiz four, you still have one more quiz, by the way. Um, know the difference between mitosis and meiosis, specifically the table that we just filled in. You should be okay. All right. So compare mitosis and meiosis, which we did already. All right. Now we're gonna move on. We're gonna talk more specifically about how meiosis happens in humans. It can happen to other organisms, right? But then in humans, we have what's called gametogenesis. Gametes, as you know, are reproductive cells, sperm and egg. Genesis is the creation. So gametogenesis is the creation of gametes, okay? Gametes are cells involved in sexual reproduction. And for humans, it takes place in the gonads, testes, in the males, and ovaries in females. Testis is singular, testes are plural. Okay, testicles means the same thing, okay? So for male, the specific process of producing gametes is called spermatogenesis. Again, it's not difficult to understand these names. Genesis, it's to create, and what are we creating? We're creating sperm. How is that different from gametogenesis? Well, gametogenesis is just the genesis of gametes in general, but if you're producing sperm, then it's spermatogenesis. If you're producing eggs in females, then it's called oogenesis. This is not a typo, it's actually double O, because the egg is called ovum, okay? And I don't know. I don't know why. I don't know exactly why it's double O, but but it is okay. So spermatogenesis is production of sperm, which is normal meiosis. Uh, oogenesis, it's the production of eggs in the ovaries, and that is complex meiosis. And I'll tell you why. Okay, side by side comparison. On the left side, we have the spermatogenesis, which begin with a germ cell, and the germ cell is called spermatocyte. Okay, how many chromosomes in the spermatocyte? 46 chromosomes, full set, 2N. Okay, and then it goes through the first round of meiosis, and it's going to be N. And then it's going to divide one more time. And in the end, you have four sperm cells, which are also haploid, one set of chromosome. Nothing is um, out of the ordinary. It's basically the same process we have been learning for half an hour. Or more than that, actually, 45 minutes now, right? More than 45 minutes. Now let's take a look at the oogenesis. Okay, so in oogenesis, we begin with a oocyte, which is the female version of a germ cell. And in the ovaries, the oocyte will divide, but this is uneven cytoplasmic division. Okay, the egg is much bigger than the sperm and it needs a lot of cytoplasm because the sperm will only be supplying the DNA and nothing else. The egg is responsible for containing all the nutrients and all the organelles that are necessary to fuel the division of the zygote and create the embryo. Okay, so when the cell divides in meiosis, you don't split the cytoplasm 50-50. Okay, instead, one cell is going to get a majority of the cytoplasm, the other is just going to get a tiny bit. So this one gets most of it, and that's going to become the egg. And this one, this polar body has little cytoplasm. Okay, and it might divide again to make more polar bodies, or it might get reabsorbed by the body. Okay? Bottom line is the polar bodies, they don't become eggs. And this thing, when it divides the second time, same thing, we have another uneven cytoplasmic division. And the egg is going to get most of the cytoplasm, and then we generate another polar body. So in the notes earlier, I said female, the number of daughter cells would be one egg, and then three polar bodies. These polar bodies, they are not going to be fertilized. They're just going to be reabsorbed by the body. But for male, they are going to get four sperms for every meiotic division. Okay, that's one difference. Couple other differences. Spermatogenesis occurs in the testes. Oogenesis occurs in the ovaries. We talk about how we start with a spermatocyte becoming four different sperm cells, but for oocyte, it will just give one haploid egg. We talk about the uneven cytoplasmic division, which is going to give you the polar bodies. And lastly, for spermatogenesis, it is a continuous process. Okay, once puberty is reached, then production of sperm is ongoing, right? And the testis can produce up to 200 to 300 million sperms daily. And if you do the math, that's as much as 2,000 to 3,000 per second. But only half will become viable. Okay, the other half is gonna be reabsorbed by the body. And for oogenesis, it is not a continuous process, right? It happens way, way, way earlier in life, before birth, right? When the baby, if it's a female, it's still growing inside the mother, the oocytes will begin the process of meiosis, but it will be arrested, it will be suspended in prophase number one until puberty. After puberty, every cycle, not necessarily a month, okay, but like, every cycle, the one of the eggs are going to be selected, and it's going to continue the process of meiosis, and it will advance all the way to metaphase number two. Okay. If it is not fertilized, then the body will get rid of it in the menses. If it is fertilized, then it will complete meiosis two, and continue to become a zygote. Okay. So rather than happening continuously like men, the process of oogenesis happened in stages. Okay. And the girl is born with all the eggs she will have in her entire lives, which is why as age as she ages, as a woman ages, the quality of some of the eggs might be affected, and that is going to affect things like fertility, as well as increased risk of chromosomal disorder in the baby, and that's something we'll talk about in a second. But any questions?
Sir, I have a question.
Sure.
Since you just talked about the eggs, so a certain number of women only produce a certain amount of eggs, right?
They are born with all they're
Certain
gonna
amount.
have. Yeah. Which
Okay.
is more than the person would need in their lifetime. Um,
Okay.
I
And
think.
as age decreases, age like you get older,
Mhm.
the egg count gets decreases, right?
Yeah, because each menstrual cycle releases an egg, right? So like you're depleting the stock, so to speak, right?
Hmm.
But the quality of the eggs will also degrade with time.
Got it.
All right. Okay, so one of the problems that could occur is what we call non-disjunction. Okay, non-disjunction. So I want to come back to here just to highlight the fact that the eggs are suspended in prophase one, and they will only progress to metaphase two in each menstrual cycle, right? Like when an egg is it's selected by the body, so to speak, right? So there's something called non-disjunction, okay? And that is a situation that occurs when the chromosomes fail to separate when they're supposed to separate. Okay? So when are they supposed to separate? Well, they're supposed to separate in anaphase one and anaphase two. So the failure could happen in either anaphase 1 or anaphase 2. Okay? So if it happens in meiosis 1, then we're going to have some problems. If it happened in meiosis 2, then we're going to have, some other problems. It's easier to show you in a picture. All right, let's start with a situation on the right side. This is non-disjunction happening in meiosis. For simplicity, I am only showing you um, one tetrad here, okay? Obviously, there are 23 tetrads, but we're just showing you one here. So in anaphase 1, this is anaphase 1 right here. Anaphase 1, which again, for female, does not happen until after puberty, right? And it's not going to happen to all the eggs at the same time, right? It's only one egg per cycle. That's going to progress from prophase 1 all the way to metaphase 2, meaning after prophase 1, it is going to be metaphase 1, anaphase 1, telophase 1, right? So it's going to happen during that time. Now, if non-disjunction happens, then the tetrad will fail to separate. The homologous chromosome that are together will not be pulled apart. Instead, one egg gets all of it, and then the other gets none. And over here, we're going to have anaphase 2. And the sister chromatids, let's say they separate fine this time. Right? So in the end, the egg here, it's going to have n plus 1 for these two. And over here, we're going to have n minus 1. What that means is the egg will have the full set of chromosome plus an extra one. These ones are n minus 1. They will have the full set minus the missing one. Okay? Whatever chromosome did not separate, then it's going to be missing right here. Okay? If the sperm fertilizes one of these eggs, okay, give me a expression here. What should I write? This is n plus 1. What should it be here? Like the math class, algebra. Someone give me an expression to describe this situation. How much genetic material does the sperm supply? How much genetic information does the sperm supply? One set, right? So the sperm supplies N. So if the sperm fertilizes the egg, then it's N plus 1, which is 2N plus 1. Meaning these cells, they are going to have two full sets of chromosomes plus an additional one. Okay? Over here, these are 2N minus 1. They will have two full sets, but missing one. Usually, missing chromosome are non-viable. Okay, the baby will not grow. Okay, to full term, there will be uh, miscarriage, for example. Over here, 2N plus 1 additional chromosomes are generally viable. You can carry the baby to full term, and then the baby will be born. But typically, there will be some disorder associated with the situation, depending on which chromosomes are extra. Okay, so people are probably familiar with trisomy 21. Does anybody know what condition is associated with trisomy 21? That's right. Trisomy 21 is Down syndrome, right? But there are other chromosomal disorder. Trisomy 18, for example, is another one. And the severity of the condition is going to vary depending on which chromosome is the extra one. Okay? So it could also happen in meiosis 2. If it happens in meiosis 2, then first division is okay. And then second division, these are okay. So we have half of the eggs being normal. And over here, this one is n plus 1. This one is n minus 1. Right? So it's less detrimental if it happens in the second half of the meiosis. because at least 50% of the eggs are going to be normal. So with age, the risks associated with non-disjunction and therefore creating these eggs that have abnormal number of chromosomes will also increase because remember, all the eggs are sitting in prophase one, and it only progressed through the division like anaphase one and anaphase two later on, right? So the longer you wait in life, the more likely that these are going to be causing problems. Okay. So how do you know if someone has additional or missing chromosomes? Well, one of the things we can do is to prepare what's called a karyotype. A karyotype is basically an orderly arrangement of chromosomes. You can extract the chromosomes from the cell and you can look at them under the microscope. You can take a picture under the microscope and you can rearrange the chromosomes in the picture. Okay, it's not like you rearrange it under the microscope or anything like that. You're just manipulating the image and you order them based on the length. The longest one is chromosome 1 and so on and so forth. X and Y kind of has its own special categories that doesn't quite fit in the length situation. But nevertheless, okay, we generally organize them based on the length from number 1 to 22. And if it's XY is a male, if it's XX it's a female. And this is a normal karyotype, only two of each chromosomes, and it has 23 in total, 23 pairs in total. Now, when it comes to genetic screening of a baby, women who are at risk. Sorry, there's a question. This is done with any type of body cells? Yeah. You can. Your cells have DNA, right? And as long as you can extract the DNA from them, then you can do it. Okay. Some cells don't have DNA though, like red cells don't have DNA, so we can use them. But you know, generally any cell is going to be okay. All right. So women who are at risk of carrying a baby with chromosomal disorder have options to screen their babies. And one option is called amniocentesis. Okay, in amniocentesis, a needle is injected through the belly button or close to there, and the process is guided by ultrasound so we don't accidentally poke the baby. And you are going to be the clinician is going to be taking a sample of the amniotic fluid. The amniotic fluid are fluids that surrounds the baby. Okay, so the baby grows in the uterus and it's surrounded by the amniotic sac. And inside the sac are these amniotic fluid that keeps the baby floating inside the mother, and kind of uh, 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 help protects the baby from external shock. This fluid contains cells from the baby, okay, that get washed off from the skin and whatnot. And so after collecting the amniotic fluid, they can extract the baby's cell and extract the DNA from it and prepare a karyotype. Okay. And in this particular karyotype, you can see there is an extra chromosome 21. So the baby has Down syndrome. And this procedure, amniocentesis, can be performed between 15th to 20th week of pregnancy. In some cases, a woman might choose to do the sampling even earlier if they are at like a higher risk, for example. Then another procedure called CVS, which stands for chorionic villus sampling, is available. So this can be done even earlier, between 9 to 14 weeks. And instead of taking the amniotic fluid, a small sample of something called the chorionic villus is collected. The chorionic villus is it's an extension of the placenta tissue, if you would, which contains the same kind of cells as the baby. And so, it's going to have the same genetic information. And then you can prepare a karyotype from that. So this one does not involve injecting any needles from the belly button, but then, there are apparatus that needs to be inserted through the cervix here and then into the womb, right? So both procedures are not completely without risk, because after all, you are disturbing the baby one way or another. Uh, but they do provide vital information for mothers to make informed decisions about their babies, should there be any kind of genetic or chromosomal abnormalities. All right. Any questions? It's like a biopsy. Yeah, not the first one though. Amniocentesis is not biopsy. You're not taking any tissue, you're just taking the fluid. This one is, yeah, similar to biopsy. You're taking a bit of a tissue. Okay, well, there are some questions here you can try later. When we come back from the break, we will do more, we'll do the second part of the lecture. Okay. What time should we come back? Let's say we come back at let's do 1:45, okay? Let's have a nice long break. See you in a bit. Okay, we are back for more. So from now on for the rest of the class, for the rest of the lecture, actually, we will be solving quite a bit of problems. You should have your study guide with you. If you haven't printed this time, make sure you print it next time, um, so you can follow along, right? So second half of the lecture is about solving genetics problems mostly. And much of what we'll be learning is based off of the work of this guy named Gregor Mendel. Has been called the fathers of genetics because of his discovery of how genetic information are passed on to the next generation. Um, we often call it Mendelian genetics. So let's take a closer look. First off, some old terminologies to refresh your memory, and then some new ones along the way. A gene is a segment of DNA on chromosome, and it contains information, instructions to make proteins. Okay? By now, you should know how to decode that information. Right? If you have the codon chart, you can convert the ACTGs into amino acids. Okay? So that's what a gene is. And genes have multiple versions sometimes, right? And these various versions of a gene we call allele. Right? So my classic example is you can have eye color, a gene for eye color, and then big B would be brown eye, whereas small b would be blue eyes. Okay? These would be different alleles for a gene. You might have more than two alleles for a given gene. For blood type, for example, the ABO blood type, there are three alleles that exist that creates different kinds of blood type. Okay. Now, when we describe a gene a person has, we look at both of their chromosomes. Okay? If the gene version, if the allele on both chromosomes are the same, then we say the person is homozygous or pure for that particular gene. Take the eye allele, for example, eye color allele, for example. If the person is big B, then we say they are homozygous, brown eyes. If they are small b, then we say they are homozygous, blue eyes. Okay? Or you can say pure brown eyes or pure blue eyes. It's important for you to stop me if you don't understand, okay? Because these are all fundamental things that you must know before we can solve the problems, okay? Now, heterozygous or a hybrid means the two alleles are different. So a person might inherit a big B from the mother who has brown eyes, and then they might inherit the little b from the father who has blue eyes. And now we say the person is heterozygous. Okay? Then you have a question like, is this person going to have brown eyes or blue eyes? Well, it turns out um, it depends on which allele, the big B or the small b, which one is dominant and which one is recessive, which is something we'll get into in just a second. So I want to show you two chromosomes here. We have the pink one, the maternal chromosome, and then the blue one is the paternal chromosome. And you can see the order of genes are the same, right? A, B, C, D, E, all the way to O, right? And some of them are different over here, so that would be heterozygous. Okay, that is also heterozygous. Now, can you find something that is homozygous? Which gene is homozygous? Yep, E is homozygous over here, or pure. Okay, anything else, guys? Yep, O, very good. That's also homozygous, right? Yeah, H as well, very good, right? Homozygous. Okay, so you guys get the idea, right? Like if it's the same, then it's homozygous. If it's different, then it's heterozygous. It's like chemistry, right? If the solution is one phase appears to be same, then we say it's a homogeneous solution. If they are not the same, if you can tell the different parts, then it's heterogeneous mixture, right? Same thing. Same thing. Okay, more terminologies. A character is a heritable feature. Okay? Something that can be passed on to the next generation, such as flower color. Remember, not everything is restricted to humans necessarily, okay? Plants can pass genetic information on as well. All living things can pass genetic information to the future generations, okay? So a character is something that's heritable. It can be passed on to the next generation. Not everything about you can be passed on to the next generation, okay? If you work out a lot and you're like very, very muscular, that's not something you can pass on to the next generation. Okay, so that's not a character in in the context of like biology terminologies. Okay, if you dye your hair purple, your kid's not going to be born with purple hair, right? It's not heritable, right? So a character is something that can be passed on. A trait is a variant of a character. So if we're talking about flower color, then the trait would be purple or white. Okay. So a character is basically something that has a genetic basis. It's encoded by a gene, but that gene, as we saw earlier, can have different versions, have different alleles. And these different alleles create various traits for a character. Okay. So eye color is a character. Different eye colors would be different trait for that character. Okay. Type of hair could be a character, and then the curly hair, straight hair, right? That would be a trait of that character. Some traits are dominant, while other traits are recessive. We use capital letter to represent the dominant trait. So in our previous example, we use big B for brown eyes. Okay, and then we use small b for blue eyes. Now, I'm sure you understand if the person has big B, then it's brown eyes, right? If the person has small b, then it's blue eyes. Okay, that's not the difficult part to understand. However, if a person is heterozygous, then is it going to be brown eye or blue eye? Well, the answer is it will be whatever the dominant trait is. So, in general, darker pigment always dominate over lighter pigment. whether it's skin color, eye color, hair color, right? So if someone inherits big B from one parent and small b from the other parent, then they are going to have brown eyes. Okay? So in order for recessive trait to manifest itself, to show up, okay, in order for recessive trait to show up, you need to have both copies of that allele. Whereas for the dominant trait, you only need one copy of that allele because it always covers up the recessive gene. Okay? Now, here is a side point that's important to remember, even though it's not like explicitly stated. If someone has a recessive trait, like blue eyes, then we know for certain what their genetic makeups are. Right? If I see someone with blue eyes, I'm like, oh, that's a little b for sure. But if I see someone with brown eyes, I actually do not know whether they are homozygous brown eyes or heterozygous brown eyes. Right? They could be big B, or they could be small b, small b. Does that make sense? I hope so, okay? Now, here are a bunch of dominant and recessive traits that you can have some fun with your family. If you're able to roll your tongue, that's a dominant trait and that's a genetic thing. Okay. If you can roll your tongue, then it's recessive. Okay. So rolling your tongue is big R, big R. You can use any letters you like. Okay. Rolling the tongue, I use big R. Okay. Or big R, small r. If you cannot roll your tongue, then you are small r, small r. Right? So listen, if the mother cannot roll the tongue, then it's small r, small r. If the father cannot roll the tongue, then it's small r, small r. And then when we say cross, that means they have babies together. Then the whole family cannot roll their tongue, right? Because they can only pass on the small r to the next generation. Does that make sense? Okay. We have someone with freckles. Freckles are like the little spots, right, on the face. Uh, this is probably Photoshop, but this looks like the same person, okay? Unless they are like siblings or something. This one has no freckles. Okay, so no freckles is recessive. So again, if both of the parents don't have freckles, then the kid's not gonna have freckles. Okay, hairline. This is called Wiggle-Peak. This is called straight, right? Wiggle-Peak is dominant. Free hanging earlobe versus attached. Cleft chin. Uh, if you're able to bend your thumb back or not, it's called a hitchhiker thumb. If you cannot do it, then it's recessive. Okay. Fun fact: if you have five fingers, five fingers, this is recessive. Okay. If you have six fingers, that's dominant. Okay. Some people do have six fingers actually. So if you really like a particular feature of yourself, like for example, oh, I love the fact that I have straight hairline and no freckles, and I want my kids to have straight hairline and no freckles. All you have to do is find someone who also has no freckles and straight hairline. Okay? Then it's guaranteed your child are gonna preserve the same trait. Any questions?
No questions, sir.
Okay, thank you. Yeah. So if you like your five fingers and you want your kids to have five fingers, also find someone with five fingers. Okay. People who have six fingers, actually, they sometimes mostly cut it off though. Okay. So you might have to dig a little bit deeper and you're were you always born with five fingers? Like, is there something I should know about? Right. Okay.
One thing
Moving
about
on. Sorry, go ahead.
one thing about the five finger, six finger. Is that a defect, birth defect, if you have six fingers?
Very
But
good
People,
question.
people,
Yeah.
people
So
feel that
there,
way.
yeah, there's actually a gene in the brain. It's called the Sonic Hedgehog gene, named after the cartoon character. When that gene gets mutated, there are some developmental abnormalities, I guess. Okay. And it could range in severity. For some people, it's just an extra appendages. It's not like the full-fledged finger. That's really rare. It does happen. It's more like a little bit of a extra pinky or extra thumb. Sometimes it can happen in the toes as well. And most people just cut it off as when they're young. Yeah.
Okay.
Was there another question? I saw someone else unmute, or maybe I imagined it. All right. So, genotype and phenotype, you will see these words a lot in questions, okay? The genotype is the genetic makeup of an organism. Are they big B, big B, or small b, right? Okay, that's the genotype. So, um, like I said, you can't always tell with certainty what a person's genotype is based on how they look. How they look would be the phenotype. Okay. So two people, two people with the same phenotype might have different genotypes. Okay. For example, big B, and uh, I said big B. Okay, big B, and big B, small b, both have brown eyes. Okay, I keep on using the eye example because it's easy. Okay, but it applies to other situation as well. All right. Who discover all these things? Well, it's the person I showed you at the beginning after the break, right? His name was Mendel, Gregor Mendel. He did experiments with peat plants, right? Peat, like little green peats that people add to their, I don't know, soup or shepherd pie or what have you. Okay, the green peats.
And then
You
he crossed thing them, right?
Yeah, exactly. He crossbreed them. Exactly.
Yeah.
Okay. So the green peats are the ones that we are used to seeing. You buy them frozen and whatnot, right? But there are yellow ones as well. There are wrinkle ones, and then some are round, and the pots can have different colors. So each of these um, characteristics are basically a character that he was able to keep track of. Okay. And by doing studies, he was able to find out each character has some variation. They have traits, different traits. So for the shape of the seed, they might be round, they may be wrinkled. For the color, it might be yellow or green, and so on and so forth. And then he figured out which ones are dominant and which ones are recessive. Okay, it's same as what I showed you in the human version. Okay, but now he's keeping track of them in plants. There are a couple reasons why he chose the peat plants to study. Number one, there are no ethical issues when you work with plants, right? You can choose to breed whichever plants you want. They're not gonna, have a problem with it. And it's easier to breed plants than it is to breed animals, right? Uh, you, I don't know if any of you grow plants indoor, right? Like in the winter, if you have plants indoor and you want to fertilize them. You just take a little Q-tip and take a little bit of the pollen and then you touch the middle part of the flower, right? And then that's gonna fertilize the plant, and then you're gonna get fruits, right, from it. And so that's basically what he did. He used I'm sure they didn't have Q-tips back then, but I think he used like a brush to collect the pollens, and then he could choose which plant to cross it with easily. And pea plants, turns out it grows relatively fast, so you can see many generations in a short period of time, and that caused the study to progress relatively fast. Okay, so to recap, he used pea plants because number one, it's easy to control which plants to cross. Number two, it's fast to maturity, and you can observe many generations in a short period of time, and probably is accessible and cheap also, right, at the same time. Okay, so that's the traits here. Um, you can keep a reference, keep this as a reference as we progress to the questions. Okay, now he had two laws, right? One of the laws is called the law of segregation. Now, I'll just read it to you first, and then I'll explain to you in plain English. During the formation of gametes, each gene separates from each other so that each gamete carries only one allele for each gene. That's the law, okay? What does it mean? Well, let's go back to the eye color example, okay? Let's say someone is heterozygous brown eyes. What does that mean? Well, it means on their chromosomes, one chromosome carries the big B gene, the other chromosome carries the small b gene. Heterozygous, different alleles. Okay. Now, during meiosis, which is the process of forming gametes, you are going to divide the genetic information by half. Okay? You go from diploid to haploid. And so these chromosomes are actually going to separate, right? And then you're going to have one gamete that has the big B, and then the other is going to have a small b. Basically, what the law says is even though you have two copies of the gene, whether they're same or different, it doesn't matter. You are only passing on one of those two genes to the future generation. Okay. The chromosomes separate, segregate as you form gametes. That's basically what the law says. But we know this already, okay? We learned meiosis earlier today. We know the chromosomes are going to separate. We know we only pass on half of it. Okay. All right. I am going to do this next part in the notes um, so that I can post it for you afterwards. All right. So this is one of his experiment. Okay. The first generation that we start observing. Okay, that's called the P generation, the parent generation, right? So he started studying one trait at a time, okay? Because it's really hard to keep track of, everything all at once. We try to, observe one variable at a time in an experiment, right? We learned this way back in September. Right? So he chose to observe the shape of the seed. Okay, is it a round seed or is it a wrinkle seed? Okay. So he took a plant that is pure round seed, pure round seed. Right? So how does he know it's pure? How does he know it's not like, big R, big R? Sorry, how does he know it's big R, and not big R, small r? Long and long answer made short. Basically, he took a plant and then he self breed the plant for several generations. And if it always produce round seed, then he assumed it is going to be a homozygous or pure round seed plant. Okay. Um, it's the same idea as people who buy like purebred dogs. Okay. They are purebred because they always consistently produce the same trait. Right? In other words, they are actually homozygous for those traits. Okay. So he took a pure round, a plant with pure round seed, which we can write big R for its genotype, and then it's going to cross it. It's going to breed it with a pure wrinkle seed, which is little r, little r. Okay. And they're gonna produce offsprings, and he observed the genotype of the offspring. So that first generation that you create from the parent generation, we call that F1. So let's take a look here. This plant has big R and big R on the chromosomes, and during meiosis, they separate the two alleles. So half of the gametes are going to have the big R, and the other half actually will also have, excuse me, the big R allele. Okay. That means 100% of all the gametes are going to be carrying the big R allele. Similarly, for the wrinkle one, it's pure small r. So there are only one kind of gametes. Okay, 50% will carry one of the small r, the other 50% will carry the other small r. Okay, so this here is called a Punnett square. Punnett, Punnett square. Uh, Punnett, sorry, square, which helps you predict the ratio and the percentage of the offsprings. Right? So let's say this gamete, big R, combines with this gamete. Then what do you get in the end? You're going to have a plan that is big R, small r. Okay? And we can repeat the process here. Big R, small r, big R, small r, and then big R, small r. So the F1 generation is, yeah, Punnett is the name. That's right. 100% big R, small r, which is 100% round. Okay, so if you take pure round cross with pure wrinkle, the first generation, all of the plants will have pure round, will have round seed rather, not pure. They will have round seed. Okay, so this shows that round is dominant over wrinkle. Okay. Now, let's go back to this picture here. That's basically how he figured out what's dominant and what's recessive. Right? So in another experiment, he probably took plants that always produce yellow seeds and then cross it with another plant that always produce green seeds. Okay, so they cross it, and he found out in the first generation, when you cross a yellow and a green seed plant, you always get yellow seed. That means yellow is dominant all the time. Right? If you take plants with peat plants, that is, with purple flower, and you cross it with peat plants with white flower, okay, and then you see that all of F1 are purple, then you know purple is dominant over white. Okay. Now this only works if the parents are pure, right? If the parents are homozygous. And again, how did he know they're homozygous? The plants would have had to produce purple flowers for multiple generations. Okay, then he could assume that is a pure plant. Uh, okay, so I just received a direct message, but isn't it possible to have traits that either parents' traits have? That's true, okay? We're gonna get to that. Keep that in mind. Okay, so let's take a look at the second part here in the F2 generation, and I think this is gonna address the question. Give me a second here. Too many windows open. This is the last thing we're gonna do, okay? Maybe. I don't know. Depends how much time we have. All right. So what he did next was that he took the plants from F1 generations. In other words, he took two plants that are heterozygous round seed, and then they breed them. Okay. So this is from F1, and it's big R, small r, big R, small r. Okay, they're all big R, small r from the first generation, right? And now we're going to create the second generation, the F2 generation. So what kind of gametes will this plant produce? It doesn't matter whether you put it at the top or put it in the side, okay? The things are going to work out the same. So for this one, half of the gametes will have big R, the other half will have small r. Similarly, for this plant, half of the gametes will have big R, half of it will have small r. Okay, for plants, it's pollen and eggs, right? Instead of sperm and eggs. Okay, so if this gamete and this gamete join together, it's going to be big R, big R. If this one and this one come together, it will be big R, small r. and so on and so forth. Let's write out the expected genotypes of F2 generation. We have big R, 1 out of 4. That is 25%. Big R. We have big R, small r, which is 2 boxes out of 4. That's 50%. And then finally, small r is 1 out of 4, another 25%. Okay, what about phenotypes? Okay, phenotypes is what they look like. It's big R, round or wrinkle. Round or wrinkle. Let's take a look at here. Round is big R, which is the dominant trait. Wrinkle is small r. So over here, big R, that's wrinkle. What about big R, small r? Is that wrinkle or round? This is round, and this one is also round, right? Because the big R, the capital letter is the dominant trait, which always shows up, right? So if you add them together, it's 75% chance it will produce a round seed. Okay, 25 plus 50. And then there's a 25% chance it will be wrinkle. Okay. Any question about the calculations? Well, we're not really calculating anything yet, but like, with the percentage and stuff like that. Okay, so that's what one of the students were asking me. The parents are both round, then how is it possible that they can produce a wrinkle seed? Well, this is possible as long as the parents are heterozygous. Okay. In other words, they have a wrinkle allele, but it's not showing up in them. Okay. But if they both pass on the wrinkle allele, then the seed is going to be wrinkle. So in this picture that I was trying to show you guys with the family, with the not the family, but the human traits, right? It's possible for two parents to be able to roll their tongue, but the child is not able to do so. Okay. In this case, the parents would be big R, small r, big R, small r. And since they each have a small r, if there's a 25% chance that they will be unable to roll. The opposite is not true. If both parents are unable to roll the tongue, then the whole family will be unable to roll the tongue. Because if small r cross small r, then it's 100% small r, small r. Okay? Any last minute question? All right. It will get more difficult for Wednesday, so please come to class prepared, okay, with the notes and the questions from the study guides, and we'll go through it together. All right. That's all for today. Thank you. See you guys next time.
Thank you, sir.
No problem.